Welcome back to the London Futurists podcast. Our subject today is the idea that the body uses electricity in more ways than are presently fully understood. We'll be considering ways in which electricity, applied with care, might at some point in the future help to improve the performance of the brain, to heal wounds, to stimulate the regeneration of limbs or organs, to turn the tide against cancer, and maybe even to reverse aspects of aging. To guide us through these possibilities, who better than the science and technology journalist Sally Adee? She is the author of the book We Are Electric, Inside the 200-Year Hunt for Our Body's Bioelectric Code and What the Future Holds. That book gave me so many insights on my first reading that I went back to it a few months later and read it all the way through again. Sally was a technology features and news editor at The New Scientist from 2010 to 2017, and her research into bioelectricity was featured in Yuval Noah Harari's book, Homo Deus. Sally, welcome to the London Futurist podcast.
Thank you so much for having me.
Thanks for joining us, Sally.
Sally, how did you become interested in this field?
I started my career at IEEE Spectrum, which is the magazine of the Institute for Electronics and Electrical Engineers, or maybe it's the other way around. It's their flagship magazine. I was on the semiconductor beat. And while sometimes that could get quite boring, I had to go to things called the Hot Chips Conference. It also let me delve into some of the algorithms that they were looking into for signal processing, for neural prosthetics, brain-machine interfaces, how they were able to read actually sort of the electrical language of the brain and decode it into things that made sense. Obviously, there's been a wild amount of progress in that. When I was doing it first, I think it was maybe like a word per minute. And now with machine learning, that's gone through the roof. They're able to decode something like 65 words a minute, I think. It's absolutely incredible. But anyway, the point was that it was possible for electrodes to read the signals from the brain. But then I started to hear people talk at conferences about electrical stimulation possibly having fairly precise effects on the brain. If you turned it the other way, instead of reading, you could write to the brain and feeding those signals back. And I realized at that point that I did not understand what this interface was between neurons and electronics. I just didn't really understand how were neurons amenable to electrical manipulation. And that is a rabbit hole that I'm still very much climbing my way out of.
You're still climbing your way out of that rabbit hole?
Oh, absolutely.
Spoiler alert, you haven't found out entirely how it works yet.
Well, there's the model that people have developed in terms of the spike and the action potential. This has been extremely well characterized. We understand how to read this language, and we understand how to process it into something that we can use. For example, like I said, being able to listen in on imagined speech in people with ALS or other disorders that disallow them to speak or move. There's been incredible progress in that. That bit is well understood. But that's the only place we've looked. There are still a lot of questions about how much further you can take that in terms of stimulating the peripheral nervous system, for example, not just the central nervous system. The fact is that the nervous system didn't invent electric signaling. Every other cell in the body uses it. But we've only looked at the nervous system because, first of all, we're very interested in our own brains. We're fascinated by our own intelligence. That's where all the interesting stuff happens. Perception, consciousness, awareness, movement, motion. That, of course, that's going to take up most of our interest. But we've kind of focused on it and possibly been not overly credulous, but we believe every time somebody tells us that they found like the neural code for how to implant a memory. But at the same time, the flip side of that, which I found really interesting, is that as soon as you get out of the nervous system and you start talking about electrical activity of cells that are not within that framework, people start getting awfully skeptical very quickly. and it starts getting associated with all kinds of electroquackery. And I think that's the rabbit hole I can't quite find my way out of, because I find it endlessly fascinating. Like, why is that?
It does sound like quite a deep labyrinthine rabbit hole to go down. I didn't know this fact that it's possible to decode 65 words a minute of human thought using machine learning.
Yeah, this was at the beginning of this year. It was a big Stanford paper. I think the best coverage came from MIT Tech Review. Antonio Regalado there writes about this stuff.
Roughly how accurate is it? I mean, if I was to sit here with whatever it is, appropriate machinery attached, could I effectively dictate a letter and email a book to a machine just by thinking about it?
I mean, it depends how you want to attach this machinery. This was a brain penetrating electrode. So you have to have neurosurgery. That's the issue with these penetrating electrodes.
Okay, well, say I'm very bold and I'm willing to have a spike stuck in my head, something a bit silly like that. Could I dictate a letter to a machine?
I imagine you could, but we speak much more quickly than 65 words a minute. So this is the sort of thing that you do when it's a matter of last resort. You have ALS.
Yeah.
And also you have to train the algorithm. It has to be trained on your particular patterns of brain data. I think it's consistent enough. They found consistent enough features that they're able to pick out these features.
It's pretty remarkable.
Oh, it is.
I think 65 words a minute would be about half normal speed. That's pretty good going.
It is. It is amazing.
Does it work in reverse? Could you have a machine put thoughts into my head?
Well, this is exactly the part where I get a little bit skeptical. There's been a lot of promise to that effect, based on the fact that we can read this pattern of spiking neurons and the sort of spatiotemporal code that a bunch of them execute together, and properly train an algorithm to pick out salient features in order to decode these things. It is absolutely remarkable. You're right. It's super remarkable.
Yeah.
But then based on that, a lot of people say, possibly without a lot of anything to back it up, that they can do the reverse. They can write the neural code. And that's where the bullshit detector stuff needs to go off a little bit more than it has, I think.
Your book has a lot of history of people being blamed as being quacks, as not being good scientists, and some of them indeed are probably exactly described like that. But there are others too whose reputations seem to have been vindicated. As a student of the history of science, I loved these episodes. It reinforces my view that the best foresight often comes out of better hindsight. Do you have any particular favorites from these stories that you tell?
Yeah. I spent a lot of time looking through history at the bottom of the British Library, and it was really daunting. And I think what unites my favorite stories is that I've used the travails of the scientists who are trying to understand these phenomena to actually try to get into their heads a little bit. And that helped me get to grips with the science. I actually got that piece of advice from Richard Panic, who does a lot of history of science writing, and he helped me with 2 of my chapters. And he said, just stand toe to toe with the scientists, or just like stand next to them and look at the world as if it was them. Don't use metaphors from the wrong century. Just pretend you're them. What information do they have at the time? The place where that really crystallized it for me was Alexander von Humboldt, looking at the work of Volta and Galvani. When he said, maybe they're both right, Galvani, that there was maybe some kind of nervous electricity in the actual biology of living things, and Volta saying, no, you have to have this dissimilar charge. When Alexander von Humboldt put forward the idea that they were both right, they weren't competing principles, that's when I started to understand it. And it was by way of looking at the experiments that he had done, which were just mad. The experiment that I describe in the book captures the spirit of my approach to the book. He took a silver wire that was attached to a galvanic stack proto battery, and he threaded it into his rectum. And he just kept pressing this electrical thing until he saw sparks and had like visual hallucinations, and he had horrible abdominal cramping. And you would think, why are you putting yourself through this? But he really wanted to understand, is there some kind of reaction that is electrical in nature? And I mean, just the fact that it didn't just affect his gut, it affected his perception. That made it really salient to him. Yes, there is something going on that doesn't just happen locally. So how is it being transmitted? That is an absolutely wild story. His friend, who participated in a lot of these horrific self-experimentations, he actually died. Some historians think that he overtaxed his nervous system. I thought that was a really great example of how far these scientists were willing to go to understand something that was such a mysterious phenomenon.
So listeners, don't try this at home.
No.
But what listeners are trying at home, at least, they are trying to zap not their rectum, but their brains, aren't they? There are many people who have got kits with the hope of pushing their brains into states of focus or creativity or mathematical genius. You wrote an article about this in 2012. Zap your brain into the zone, fast track to pure focus. Do you think there will be a future when more of us are doing that?
FDA in the US hasn't approved anything for these indications. They are wary, probably correctly, of a long tail of electroquackery. But I want to decompose what you said a little bit because it really depends on what you mean by brain performance. This question is the same across a wide variety of different scientific pursuits that's going on right now, from gene editing to longevity research. Where's the line between enhancement and therapy? Can we improve our brains with electricity, being the question. So for therapy, the answer has been yes for a long time. DBS, deep brain stimulation for Parkinson's disease. That's been a slam dunk. Then just this week, there was an article about a brain implant that rescues memory in people with traumatic brain injury. There's a researcher at Oxford, I believe her name is Charlotte Stagg. She looks at stroke recovery using transcranial direct current stimulation. Other trials, like last year, there was something where non-invasive brain stimulation helped older people improve their memory. The whole point here is things start to get really blurry between enhancement and therapy. And this is something that I really encountered while I was thinking about what happened to me in 2012. Because for me, at the time, it felt like I had become some kind of super sniper. I felt mentally improved.
This is a story you tell at the beginning of your book and revisit at the end of your book.
Yes.
We won't spoil too many things for readers, but it is quite eye-opening or jaw-dropping.
What I found really interesting was that I kept, during the next few years, not just for my book, but just generally, anytime I would meet someone who did TDCS research, I would buttonhole them and I would tell them about the experience and I would be like, what happened? And more, I started to realize that I had maybe hallmarks of, I don't know, anxiety, depression, ADHD, I don't know what it's part of, but angry little voices that were always putting me down inside. And the TDCS made those go away. I noticed that at the beginning.
I haven't read your account of what you did. So how much intervention did you have in order for these effects to occur?
Oh, not much. I had a, not homemade, but lab homemade, 3D printed electrode, cathode and anode, and then a little 9-volt battery basically hooked up to me. And there's a whole preparatory thing. There was a task that I was doing over and over again and just remarkably not getting better at. And then I had the transcranial direct current stimulation. It was 20 minutes. And I think I got lucky, to be honest. First of all, the stuff that you get as a consumer device bears very little resemblance to what I was hooked up to. I had been chasing these people at DARPA for years to let me try it myself because they have a thing. You can't really interview soldiers who have had this. They have a real ethical line where they don't want people like me exploiting soldiers for articles in the media. So I basically had to resort to do it myself.
Sure, but if there's reasonable evidence to suggest that you can make people effectively smarter or more competent in a significant way using this technology, then I'm surprised it isn't more widely researched, more widely used. And frankly, I would like it to help me learn Spanish because I'm old and I'm very slow at learning Spanish. I could do with some TCDS to help with that, I think.
Did you know, actually, there is a lot of evidence that part of the reason we learn less well as we get older is because of those voices. We just have these expectations that are completely unrealistic. So then we just have our little ass-kicking machine that we use to chase ourselves around instead of learning it properly. Anyway, the point is, there has been a load of research into TDCS. And the thing that gold standard meta-analysis found in 2016 is that we don't have the parameters exactly right in order to create a device that works for everyone. First, because the device manufacturers, they just want to ship something that sounds plausible. And been a lot of these TDCS devices, they're largely probably not very effective. But for being in a lab, You have to know very specific details about the person because electricity is not unlike a drug. It isn't like the old quacks used to do, just step 1, zap, step 2, question mark, step 3, efficacy. You have to know parameters like what's the thickness of the person's skull bones. I know that sounds crazy, but it is that specific. And you have to get the device to the point where you can actually program in that kind of variable. Otherwise, it's a crapshoot. If I hadn't gotten lucky, I guess my skull is of a certain thickness. I don't know. But, because bone is electrically conductive.
Yeah.
A lot of the studies that have sought to debunk the fact that TDCS could possibly work at all have said that most of the electricity just gets siphoned off. They're still hammering out what is the exact mechanism. Does it actually cause action potentials, or does it make the neurons more likely to fire in synchrony? This is a really hot topic. It is very much under investigation. They're getting places. They know, for example, now that for depression, this has fairly robust evidence that this works for depression. For stroke, there is good evidence.
In your case, having had a significant positive effect, I would imagine you would have been chasing DARPA or whoever gave you this thing in the first place to have some more.
Yeah.
Have you been doing that?
I did initially. I went to the lab of Roy Cohen-Kadosh at Oxford. He does experimental neuroscience and experimental psychology. I made him kit me out so that I could do a bunch of maths tests, and it didn't work. So I was like, wait, what? Did I fall for a placebo effect? This is why I've been down this rabbit hole so long, because actually it was a completely different setup, a different electrical setup. I think I was getting a different amount of amps. These are all the little nitpicky things that you've got to figure out.
Yeah.
If you're going to design something that works for people. We're down in the trough of disillusionment / basic research, building our way back up.
Right.
I don't know who we is, but yeah, I want a device. I do.
Yeah. So, David, can I break the podcast's fourth wall, so to speak, and ask you a question? Because when I used to go to your meetups when I lived in London, there was I think he was Russian, or an East European guy.
Ukrainian,
I beg his pardon.
Andrew Vladimirov. You both spoke at my anticipating 2025 conference in 2014.
Yes, that's right. So he was very, very keen on TCGS, and he was using it on himself a lot. Is he still doing it? Is he successful? Is it producing positive results?
Well, I think it helps him a bit, but it doesn't make him reliably superhuman all the time.
Right. Okay.
It bears, I think, what Sally has said.
There's a long way to go.
It still needs investigating.
Yeah, it's a calibration issue.
But maybe we should bring Andrew onto the call at some stage as well.
This is also where I always bang my drum, that it's not all about the nervous system. There was a series of debunking meta-analyses in I think 2014 to 2016 where people were just like, oh, this is all bullshit. And here's how we're going to prove it. Some people put TDCS on a cadaver, and they wanted to prove that there was not enough that got into the brain to generate an action potential. This is where things get really thorny because, number one, we don't really think anymore that the brain is this straightforward computer thing where you're just like, I will make an action potential and then things will happen. The electric field has other subtler effects. And also, bone cells are electric just as neurons are electric. Just in brief, the meta-analyses showed that nothing happens, but better studies have been done since then. And they show that there are effects on things like depression, which I think is really interesting. But you have to calibrate to individuals. And also clinicians should calibrate the actual parameters that'll work for people. I don't think it's going to be some kind of one-size-fits-all silver bullet. It'll be something that has to be personalized.
We'll be right back after a quick break.
The People in Performance podcast offers insights into the strategic competencies, behaviors, and results needed to increase the performance of employees and leaders. The show looks at all areas of the world of work, including attraction and recruitment, onboarding, training, and succession planning. Tune in to get a better handle on the stages of workforce planning you need to master in order to guarantee long-term success in a rapidly changing world of work. New episodes drop each week. Check out the latest People and Performance episodes on your podcast platform of choice. Follow us on socials and read the latest articles at pppod.com.
Let's talk about cancer. There are some suggestions that the reason cells become cancerous is because of a breakdown in the normal exchange of electricity between neighboring cells. There are some suggestions that some electrical interventions might undo some cancer. Now, we don't want to get people's hopes up. The technology is immature. Some proponents of new treatments have been, let's say, economical with the truth. But your book does tell some positive episodes. How optimistic should we be?
Well, there are already some really interesting treatments in the clinic. There's something called tumor treating fields, TTF. These are mild electric fields these are used for glioblastoma. You wear, I think, a funny helmet looking thing, and the fields pulse through the skin of the scalp, and they interrupt cancer cells' ability to divide. And what they're doing is they're taking advantage of the cell cycle, which is something that I just want to put a pin in right now, the word cell cycle. I want to get back to that because that's strongly bioelectrically mediated. But basically, they're taking advantage of the fact that, broadly speaking, cancer cells divide like crazy. Neurons that are healthy don't divide. So if you can hit something that is in the middle of dividing with an electric field that interrupts its ability to divide, then that's keeping a tumor basically in place. You're interrupting its ability to spread, whereas you're not interrupting neurons' ability to do very much because they're not dividing. So there's been some really interesting papers about TTF and glioblastoma. More excitingly, they applied tumor treating fields to lung cancer. There was a paper in The Lancet in September that showed that this could actually enhance the efficacy of existing drug regimens. I'm not sure exactly how it works for that because that's quite new, but it's really exciting that you have these electrical interventions that seem to be proliferating. There's also something called room temperature plasma, which is basically like they figured out how to wield room temperature lightning. And they've been using that in cancer as well. So there's a lot of, you might not understand the mechanism precisely, but you are starting to see real effects that are pretty indisputable. And then there's the people who are looking at why this might be working. And a lot of their stories is what I told in the book, where they're starting to understand what happens to a cell when it goes cancerous. And this is where the cell cycle thing comes into play that I wanted to bookmark earlier. So the cell cycle is basically where the cell differentiates. It goes from immature to mature. It assumes its job in your body. So you go from an undifferentiated cell to like a neuron or a skin cell or a skeletal muscle. And as it does that, it goes through a whole lot of genetic stuff and mechanical stuff, but it's always attended by a change in voltage, membrane voltage. And they have characteristic ones, like musculoskeletal tissue is going to be something like minus 90 millivolts, which is very robust. Nerves, they are minus 70 millivolts. Fat cells are about minus 50 millivolts. I'm sure this is wrong, but I like to think of it as like different kinds of batteries. It's almost like AAA batteries, AA batteries, D cells. There's been a lot of really good work showing that if you just interrupt a cell's ability to attain that voltage shift, you totally mess with its ability to become whatever it is it's going to be. There's loads of really good gain of function and loss of function studies that bear this out. But then the interesting thing is when a cell that has a job in your body and it's trucking along and it decides that it's going to go cancerous, it shrugs off its electrical identity along with its job in the body. And it's like, you know what, screw this. I'm going to just eat whatever I want and proliferate and do whatever the hell I want. I'm not going to do this stuff in your body anymore. And at that time, it depolarizes and goes back. And again, there's been loads of amazing work that shows that if you interrupt its ability to shrug off its electrical identity, you also inhibit it from becoming cancerous or exhibiting cancer-like behavior. And they've done this in cell culture. They've also showed this in living creatures. tadpoles. And then the third wing of that, or the third leg of that research, I guess, is what is that doing? What does it help you do to shrug off the electrical identity? And what Mustafa Jamgaz, who was at Imperial College London, showed in the 2000s to 2010s is that once you have a differentiated cell, like a skin cell, it shouldn't be spiking. It shouldn't be sending neuron-like signals. But when they go cancerous, they send these signals. And that appears to maybe be part of the reason that they're able to coordinate. Cancer cells can coordinate in order to metastasize and in order to move throughout the body and consume more. There's really interesting work now looking into why exactly they do that. Like, what's the ion channel that helps them do that? And can we interrupt it? I'm waiting with bated breath. I think there's going to be some interesting work out fairly soon.
Ion channels are little holes between cells that pump out ions. It's not electrons. It's not the same kind of electricity that runs through our computers. It's positive charged ions, mainly like sodium ions or potassium ions.
And negative chloride. Basically, cells have their electrical identities because of their concentration of different kinds of ion channels on the membrane. Those guys let in certain amounts of charged particles and keep others out, which accounts for whether they become a D cell, a AA battery, a AAA battery. This is a terrible metaphor, but it's good enough, I think. That's how they maintain their voltage potential that is in charge of their identity, or rather that seems to have a lot to do with their identity.
Another big picture is that biology has been dominated for the last 70 years by the idea that genes control everything. The breakthroughs of Crick and Watson and Rosalind Franklin and many others. But we've understood in the last few decades that genes do different things in different cells. That's because of what we now call epigenetics, the things that get added onto the DNA so that it folds up differently in different cells. Well, your book raises the possibility that what controls the epigenetics is, ta-da, electricity. But if you could change the electricity in the right way, then you drive the epigenetics, which in turn drives creation of things like eyes on frogs' behinds or tadpoles' behinds. That seemed to be an area that deserves a lot more attention. So it's tied up with the idea of an electrome, which is that rather than just looking at the genome or the epigenome, we should be studying this new field, the electrome. Is that a fair description?
Yeah, definitely. The word electrome has been popping up much more now. I know that when I was interviewing people for the book, a lot of the researchers said that they had been seeing the word more and more in papers that were out for review. People are trying to get their heads around this and try to get to grips with how does bioelectricity work in biology? What is its relationship with all these other ohms? That's the part where things are still brand new. In 2006, a researcher called Min Zhao, who studies bioelectricity, but at the time was really focused on how cells move under the influence of an electric field. He got together with a geneticist called Joseph Penninger, and they found two particular genes and gene regulatory networks that were turned on by the application of an electric field, which caused them to move toward the cathode of the electric field. In the years since, some people are really looking for what's the sensor on a cell that isn't a neuron, because we know how neurons sense voltage. They have literal channels that sense voltage. But other cells that aren't neurons, how do they sense an electric field, which they do? And they can't figure this out. So there's this hunt for like the rhodopsin of electricity.
What's the rhodopsin?
Rhodopsin is what they put into cells in order to sense light. It's something you do in order to be able to do optogenetics. And optogenetics is you put a whole different kind of sensor into a cell, which allows you to control the cell using literally light to activate them. So you can engender an action potential just by using light. Anyway, it's a really, really important research tool. People talk about optogenetics a lot, but if you wanted to have some sort of light-controlled optogenetic thing in you, you'd have to do gene therapy, and people are really resistant to that. So most optogenetics takes place in mice and other model animals. Anyway, the point is that they are trying to find something that can very easily be manipulated and a mechanism that shows you how does biology do this. But the problem is that since they found those two things in 2006, they found loads of other genes that get turned on. And essentially, it's a huge panoply of stuff that gets turned on and gets changed by an electric field. At Texas A&M, they have suggested that we start a field called electromics so that we can actually map all the stuff that gets affected by electric fields. And then we can figure out how to tune them specifically so that we can create specific changes, because nobody's using the same exact setup. Nobody's using the same kind of parameters. They're not using the same voltages. It's just been really disorganized. And like the whole field needs to just get more organized to start to be like, what particular frequency turns on this gene, for example. And so that's what they're trying to start to do now, because it's all been phenomenological. We've seen these cool things happen, and then we're like, electricity works. And it's like, well, what kind of electricity? What voltage? What amplitude? What kind of stimulation frequency?
Electromics, you heard it here. Not electronics, electromics. One other thing we might touch on is the future of bioelectricity may be biology, rather than having metal pins stuck into our head, which the brain rejects fairly quickly. That was fascinating to me as well. Is that something you're studying more deeply?
Yeah, along with all the other stuff I have. I'm trying to get to grips right now with anthrobots, which is the next generation of xenobots.
Anthrobots and xenobots.
Have you seen this? The people at Tufts University who had created these little biological robots out of skin cells of frogs, they've now taken human cells to do the same thing. They take a cell, maybe a single neuron or something, and then they do this bioelectric tweak to it, which I still don't understand what it is, but this is what they did to the xenobots as well. And then the cells, they are separated from the bioelectric environment of the body that keeps them doing their job as skin cells or whatever other cell. And when you separate them, when they proliferate, they create these little blobs that race around and act like they become completely new organisms that have no genetic history on this planet. The researchers are saying that what they could end up doing with these things is using them as implants, basically, or things that you would inject into your body to eat the plaque that lines the arteries, or drug delivery vehicles. But these would be things that you could use instead of the sort of synthetic or metallic approaches that have been championed otherwise. That's one thing. But the other aspect of that is that they're really trying to move toward using the electrical properties of biological matter to try to design implants that interface better with our biology. There was a guy working on something called a proton transistor, which instead of using metal to conduct ions, would use things like shrimp chitin to conduct proton gradients. Basically, what it would do is it could obtain a signal without the damaging effects of having a piece of metal in your body.
So everything that transistors could do in principle with silicon or germanium or whatever, instead these proton transistors could do, but with protons, which are hydrogen ions.
Right, exactly. There's a lot of people looking at how to take better advantage of the way that the body actually deals with its own kind of endogenous electricity. I think my favorite example is there's a guy who is working on a battery that would power an ultra-low power implant. Instead of it being a lithium ion battery or whatever, it would actually harvest electrons from the glucose in the cerebrospinal fluid. This sounds nuts. This sounds like something from the Matrix, but they've made progress. They've been working on it since 2012. And so I think the move is, I just don't think you're going to get that much further with traditional implants because they just are so mismatched with our biology. In the brain, of course, they cause glial cells to swarm in and form really thick scar tissue around the implant, which makes it unusable after a while, which is why you have to get it either replaced or taken out after 10 years, probably. But using more biological materials will make the body accept these interventions. I think this is many, many years in the future. But if we want to keep going with implants that are supposed to help our memory in the long term, and things that you want to implant into people not toward the end of their life when they have catastrophic diseases, if you want people, healthy people, to accept implants, you have to make sure that they last their whole lives, or that they're biodegradable enough that they don't cause any kind of second order effects.
Yeah, it sounds like the sort of thing that's phenomenally complicated, and you would think, oh, this is going to take 100 years to sort out. But of course, the exponential improvement in compute and with AI getting smarter and smarter, things can move much faster than you might think. And some of this really science fiction stuff might be available to use within the lifetime of people alive today, maybe even the people on this podcast.
Oh, that would be nice.
Certainly you, Sally. I don't know about me and David.
Thanks so much for joining us, Sally. We've covered a lot of ground, but we've only, in a sense, touched the surface. So I do recommend people look out for We Are Electric.
Thank you.
Are you writing We Are More Electric as a follow-up?
Well, actually, yeah, I'm trying, but it's all going very slowly. I'm actually working on a Substack. I'm about to start a Substack because I've gotten so much awesome mail about my book, and they've had so many questions. It's just more rabbit holes. I'm in the middle of writing an answer for somebody who asked why bioelectric interventions were able to allow the creation of planarians that had two heads, and one of them being from a different species, because that was something I wanted to get into more in the book. But then I was like, oh my God, this is so complicated. And also my publisher was like, please stop writing more words. We need less words, not more words. But now, yeah, so all those extra words are going to go in my Substack.
So if people want to find out about the two-headed planarium, in which the two heads are from two different species, remarkably,
yes,
they should look for your Substack when it's available.
Oh, thank you. Thanks so much for having me on. I really appreciate it.
Thank you very much, Sally, for joining us. It's been a real pleasure talking to you.
Thanks a lot, Sally.